The sequence of amino acids in a polypeptide is dictated by the information in a gene. The DNA has a sequence of nucleotides which is transcribed into RNA and then translated into protein. The translation of that gene into a protein requires the placing of amino acids in a particular sequence. If this pipe cleaner represents the primary sequence of this amino acid, if this pipe cleaner represents the primary amino acid sequence, then if you remember from our lecture, one end is the carboxyl end and the other end will be the amino end. Now, based on that analogy, when you look at this pipe cleaner, hanging from this pipe cleaner facing down will be all the R groups. Now, the R groups are of different types. You have hydrophilic ones, you have hydrophobic ones, you have potentially charged ones, uncharged ones. Whatever they may be, these R groups, as well as the amino groups and carboxylic acid groups of the amino acids that are bound together, basically form interactions. And these are intermolecular interactions, and they are things like hydrogen bonds. And this hydrogen bonding between these molecules allows these molecules to fold. And the folding of this amino acid sequence, which is the primary sequence of a protein, is in two forms in the secondary structure. So I'm going to build it. This is an alpha helix, sort of like a helical structure. And then a beta pleated sheet is sort of the pipe cleaner folded back upon itself like this. Okay? Now, when you look at it, you have a beta pleated sheet and you have an alpha helix. And an alpha helix is a helical structure. Now, this is your these are the two possible secondary levels of structure. You can then fold this molecule upon itself, and you can see that when you fold it upon itself, it's sort of loose. And what when this secondary structure folds upon itself, this is called the tertiary structure. And you can see that the tertiary structure is kind of movable. In order to hold this tertiary structure in place, we have what are called disulfide bridges that are formed by cysteine bonds that hold this like this. And this is sort of like a completed example of a tertiary structure. Now, the fourth level of structure is a quaternary structure, and that involves more than one polypeptide. So I'm going to really quickly create another polypeptide chain. So here is my first one, the one that I made initially. And I'm going to make another one. This one I'm going to make a little different. I'm going to have some beta pleated sheets, right, in the middle, and then I'm going to have some alpha helices at the end. So this is what my secondary structure is going to look like right there. Okay, so my secondary structure, it has some alpha helixes at one end, beta pleated sheets, and then that's my alpha helix again. I'm going to fold this molecule again, and I'm going to form a tertiary structure like so. So here is a second tertiary structure. Here is the first tertiary structure. In order to form a quaternary structure, this one molecule has to interlock or form an interaction with another. And this is a protein. And a protein is made up of two or more polypeptide chains which are interacting with each other.